All right, the next group of biomolecules that we're going to talk about are proteins. Proteins basically do everything. So if you took the water out of you, at least 50% of your dry mass is made of protein. And they have all kinds of different functions. So just a few examples to run through. One of the most important groups of proteins that we're going to talk about are enzymes. So enzymes are proteins that catalyze chemical reactions in our cells. They do just about everything our cells do, okay? There are important proteins that make up our immune system. Proteins play the role of storage in things like seeds and eggs. And of course, when we think nutritionally of protein, we think of muscle, which provides energy to us as food, but for the animal, that provides movement. So proteins help move things in all kinds of different ways in our cells, whether it's moving things across cell membranes, structurally moving the whole organism, or molecularly moving small things within the cell. Proteins can also be signaling molecules, whether those are hormones or the receptors that receive signals. And of course, proteins can be structural, whether that's connective tissue like collagen or the keratin that makes up our hair and nails. So proteins are incredibly varied in their functions, but they all have the same underlying basis for their structure. Our building block for proteins are amino acids. So an amino acid is built on a central alpha carbon, and on one side it has an amine group, a nitrogen-containing group, and on the other side it has a carboxyl group. And then on that alpha carbon, there's room for two more bonds. So one of them is just filled up by a hydrogen. And then the other one is the R group, or the side chain. And it's a side chain that distinguishes one amino acid from the next. So this NH2 group is the amine group. And the C double bond OH is our carboxylic acid. And that's where we get the name amino acid from. You'll see amino acids written like this, or you could see them in their ionized form, where it's actually NH3 and COOH minus. Okay, so that hydrogen group can break off, and this double bond actually can flip positions. So you might see it with a negative charge here, and you might see it, like this, or you might see it with a positive charge on the amine side. So our amino acids are our monomers for proteins. Just like with most of the other biomolecules, our monomers are going to be joined together to form polymers. So here I've got two amino acids, and just like with the other molecules we've talked about before, they're going to be joined together by making a new bond after making a dehydration reaction. So the hydroxyl group from one and the hydrogen from the other are going to leave and form a water molecule. And the new bond that's formed is going to be between the C double bond O in one amino acid and the amine group on the other. So this new bond that's formed is called a peptide bond. You should be able to recognize the structural formula for an amino acid, and if you're looking at a chain of amino acids, you should be able to determine which carbon is the alpha carbon in the center of the amino acid, and which carbon is the carbonyl carbon, and where the peptide bond is. So a protein is going to have a repeating backbone of amine, carbon, carboxyl, peptide bond, amine, carbon, carboxyl, and then if we had another amino acid down here, this would be the peptide bond, and then another amine, carbon, carboxyl, and so on and so forth. Okay. So you guys should be able to see this. You should draw it out to practice it. So filling in our notes in our study guide, our monomers are amino acids. Our polymers are called polypeptides. They're called polypeptides because the dehydration reaction that joins them together forms a peptide bond. All right. So if you just have a chain of amino acids, if you just have a polymer of amino acids, you have a polypeptide. But if you had 1,000 alanine amino acids joined together, that wouldn't do anything. It would be a polypeptide, but it wouldn't have a function. A protein is a polypeptide that's folded into a specific three-dimensional shape that allows it to have a specific function. And so we have to talk about how and why proteins fold up so we can understand the shapes they take on and the functions they have. Part of the how and why for how proteins function is what particular amino acids are present in the protein. And the difference between one amino acid or the next is the R group or the side chain. So in this image, the repeating part of the protein, the amine, carbon, carboxyl, is highlighted in gray, and then the R group, the side chain that's unique, is highlighted in different colors. Now, you don't have to memorize the structure of all the different amino acids until you take biochemistry, but what I want you to notice is that these R groups or side chains have different chemical properties. So all of these up here are mostly made of carbon and hydrogen, so they're non-polar. Whereas these down here have hydroxyl groups or sulfur-containing groups or amine groups that make them polar and hydrophilic and mix well with water. And there are also acidic residues that ionize or basic residues that ionize that can form ionic bonds. So we're going to see that different amino acids are going to cause the protein to fold up in different ways. There's actually 4 different levels of protein folding that you guys need to know about. And just to start off with, they're just called first, second, third, and fourth. But of course, to make things more complicated, we're going to call first, second, third, and fourth slightly different things. So the first level is called the primary structure. The second level is called secondary. Tertiary is just a weird word for third, okay? And then quaternary is the 4th level of protein folding. So we're going to go through each of these. All right, so the first level of protein folding is the primary structure. The primary structure is simply the order of amino acids. And so what defines the primary structure is actually the information stored in our DNA, in our genes, okay? And if anything changes what amino acid comes next, then you've actually changed the primary structure. Primary structure is held together by the peptide bonds that join each amino acid to the next. So primary structure, what it is the order of amino acids held together by peptide bonds. All right, so we initially have this linear chain of amino acids, but those amino acids are going to start to fold up. And the first true level of folding is the secondary structure. And there's 2 basic shapes that we're going to see. Alpha helices or a beta pleated sheet. The important thing that you need to know about secondary structure is what holds it together is hydrogen bonds along the backbone. So polar parts of the repeating part of the protein are going to hydrogen bond to each other. So if we look down here at this beta pleated sheet, we have blue is nitrogen, black is carbon, red is oxygen, and then the R groups are these white balls, and then gray is hydrogen. So we have amine, carbon, carboxyl. Here's a peptide bond, amine, carbon, carboxyl. Here's a peptide bond, amine, carbon, carboxyl, and so on and so forth. And for secondary structure, it's hydrogen bonds between the positively charged amine groups and the negatively charged carboxyls that are attracted to one another. Those opposite partial charges attracting hold them in either a beta sheet or an alpha helix. Now, whether it folds into a beta sheet or an alpha helix is influenced by what these R groups are, but it's not actually bonding through the R group that forms the beta sheets or the alpha helices. The next level of protein folding, tertiary structure, is where those R groups come in. So tertiary structure forms the 3-dimensional shape of the protein. And it's accomplished through bonds through the R groups. And every different kind of chemical bond that we talked about can contribute to tertiary structure. So you can have hydrogen bonds between 2 R groups, or you can have a hydrogen bond between 1 R group and the backbone. You can have hydrophobic residues hiding from water getting pushed together and kind of sticking by van der Waals interactions or hydrophobic interactions. You can have ionic bonds formed between basic and acidic residues, and you can even have covalent bonds between sulfur groups that are strong permanent bonds. Okay. So tertiary structure is the three-dimensional shape of the protein that's held together by all the different kinds of bonds that we talked about through the R groups or the side chains. And then our last level of protein folding is quaternary structures. So not every protein has a quaternary structure. Quaternary structure is when multiple subunits join together. Again, every kind of bond that we talked about can contribute to quaternary structure, but it's primarily held together through weak interactions like hydrogen bonds and hydrophobic interactions. But it can also include ionic bonds and covalent bonds. So the example here on the slides is hemoglobin. Hemoglobin is the protein in our blood that carries oxygen. And it's actually made of 4 separate subunits. Each of these subunits by themselves would be a polypeptide. They don't work on their own, but when they assemble into their quaternary structure, now they can do the job of carrying oxygen in our blood. Anything that disrupts these different levels of protein folding is going to cause the protein to unfold. And if you lose the shape of the protein, you lose its function. So if you change the pH, it's going to change the hydrogen ion concentration in the solution, which can disrupt hydrogen bonds and ionic bonds. And if it's a strong enough acid or base, it can even break covalent bonds. If you change the temperature, the first bonds to go will be the hydrogen bonds, which is going to unfold the secondary structure and the tertiary structure. If you put it in an organic solvent, that's going to allow the hydrophobic residues that were buried on the inside of the protein to flip to the outside. Or if you add salt ions, that's going to disrupt ionic bonds and hydrogen bonds as well. So if you lose the structure of the protein because it unfolds, that's called denaturation. So if you lose the structure, you lose the function. And that's a vocab word you guys need to know. So proteins can actually fold up on their own. The information for how the protein should fold is actually stored in the primary structure, the order of amino acids. But to fold proteins efficiently, our cells have proteins that assist in this folding. They're called chaperonins. So they're chaperones. They're supervising the protein's activity, making sure it folds properly. And there are diseases associated with misfolded proteins. In Alzheimer's disease, there are these plaques of this tau protein that clumps together because it's misfolded and disrupts neuronal activity. There are infectious proteins called prions that are misfolded proteins that cause other proteins to misfold, which also disrupt neuronal function. And this causes mad cow disease in cows or Creutzfeldt-Jacobs disease in humans. One major group of proteins that we're going to talk about more in the next unit are enzymes. So enzymes are a kind of protein, but not all proteins are enzymes. Enzymes are a kind of protein that are biological catalysts. They speed up chemical reactions without being used up or altered by the reaction themselves. So one example of this is lactase. Lactase is the enzyme that breaks down the disaccharide lactose, and form fits function. So lactase can only accommodate lactose. It can only break apart the glycosidic bond in lactose. If you tried a different sugar like sucrose, it doesn't fit in the active site of the protein and it can't break it apart. We'll talk more about how enzymes function in the next unit. So let's make sure that we have all the information in our notes. So our monomers of proteins are called amino acids. Our polymers are called polypeptides. Our monomers are joined together by a dehydration reaction to form a peptide bond. There are lots of different examples of proteins, and whether they're hydrophobic or hydrophilic is determined by the R group.